So today I will be so now I will be actually talking about the Clostridium perfringens. So actually the gas gangrene, it is actually the majority the gas gangrene, the majority organism that is responsible for the gas gangrene. It is the Clostridium perfringens. Okay, so to treat such conditions, it is very important to know exactly which organism is causing the gas gangrene because for the medical treatment. So at first we will be studying about the Clostridium perfringens general properties. Then I will go into how we can identify the Clostridium perfringens, and then a few words about gas gangrene and the food poisoning by certain enterotoxins that are produced by this Clostridium perfringens, and how we can treat it by using antibiotics. So this I will be explaining in this class. So starting about the Clostridium perfringens, as here you can see that gram staining, it is a very important, right? Because we can actually narrow down. For example, this Clostridium perfringens, they are actually gram positive bacilli. So they are they will actually appear violet in gram staining. So here you can see they are actually a gram positive bacilli. Apart from that, you cannot see any flagella here because they are actually non-motile, because there is no flagella. But most of the species that are belonging to this Clostridia, they are actually having flagella, and that flagella is of peritrichous type. So this is actually peritrichous type. So if the flagella is arranged like this, that is actually the peritrichous flagella. And you can see that here there is a capsule. That is a capsule of the Clostridium perfringens. And remember that most of the species that are belonging to the Clostridium, they don't have any capsule. But the Clostridium perfringens is having capsule. And one more thing, they are actually strict anaerobe. Okay, they are actually strict anaerobe. So they are actually found in the soil, in the actually GIT of animals, humans. So when actually they are having the spores, and these spores, actually there are many types, and the spore can be actually present. This is a spore, and this can actually be present in the center, or it can be present in the terminal end, or that spore which are round in shape at the terminal end, it can be oval, or it can be in the subterminal as like this. In the case of Clostridium perfringens, it is of subterminal type. And one more thing, the Clostridium perfringens that are actually taken from humans, that are actually grown in the humans, they don't contain this spore. Actually, when you are culturing, you cannot see the spores. There is no spore. So that is a brief idea about the Clostridium perfringens. So now going on to the identification of the Clostridium perfringens. So one is the Naegler's reaction. So what is this Naegler's reaction? So you have to take a media, that is egg yolk media, and you have to take this Clostridium perfringens and give a streak. So what you can observe is that, so just before that, I just want to introduce to you something about the virulence factor of the Clostridium perfringens. So there are actually 16 virulence factor. There are actually 16 virulence factor, and it can produce a major virulence factor as well as a minor virulence factor. So the major virulence factor are alpha, beta, iota, that means I, and the epsilon. Whereas the minor virulence factor are the theta, gamma, delta, such that. So they can actually be cultured in the Elna's medium. Elna's medium. Actually, anaerobic medium are required, and you can also even culture at a Robertson cooked meat media. And what you can observe is that in actually culturing at the it's an anaerobic medium, and so there is a characteristic feature of this Clostridium difficile is that when you are actually culturing in the Robertson cooked meat media, actually when it can break down the protein of when it can break down the protein of that meat, that is when it has a property of actually this Clostridium, okay, when it is having the property of proteolysis, the meat will turns into black color. So when it is having saccharolytic activity, the meat will be turning into a pink color. So the Clostridium perfringens, they are actually belonging to this pink color. They are actually mild saccharolytic activity. So that is why they will have a pink color. And one more thing is that they can actually in the identification of here you can see the heat tolerance. In the identification of this, it is an heat tolerance at 45 degrees Celsius heating. Actually, only this Clostridium perfringens will survive, whereas all the other species it cannot survive. So you can actually select the Clostridium perfringens from others. So what about this Nagler's reaction? So Nagler's reaction is you can take an egg yolk media as you see right here, and actually you can divide into a plain medium, and in this side you can actually add anti-alpha toxin. So basically when you are giving a streak of this Clostridium perfringens, so that Clostridium perfringens is having alpha toxin, right? So this alpha toxin, it will be actually neutralized in this side, but in this side, as this alpha toxin, it actually produce something like an opalescence here. There will be an opalescence. It is because the lecithinase, actually alpha toxin is having the property of lecithinase activity, so it actually create an opalescence. Now the second method that you can use to identify this Clostridium perfringens is by target hemolysis. So in the target hemolysis, you can take a blood agar. So before I just go into here, you can actually culture the Clostridium perfringens in, as I have earlier said, in the Robertson heat cooked meat media. Then you can use even the nutrient agar, blood agar. So here you are actually culturing in the blood agar. So what you can see is that here you can see two zones of hemolysis. So you can see that the central narrow zone, it is actually produced by theta hemolysin. That is a minor toxin. It can produce hemolysis, and the outer broader hemolysis is incomplete hemolysis is produced by alpha hemolysin. It is a major toxin, alpha hemolysin. So that is about the target hemolysin. So just a few words about alpha hemolysin is that, so it is having this alpha toxin, it is having hemolytic properties, plus they can actually causes platelet aggregation, okay, they can actually cause platelet aggregation. So majority actually all of the strains produce this alpha toxin. Then by the spectrum of actually some will produce this beta toxin alone or iota toxin, that means I, or the combination of these. So in that basis actually we have classified this Clostridium difficile into 5 groups, that is A to E. Okay, so that's about the virulence factor. And there is actually something that is called as hot cold phenomena. So what is this hot cold phenomena? So basically, as you remember in the Staph aureus, the beta hemolysin, in the same way this alpha toxin. So alpha toxin is actually they will start their sorry the actually the theta. So they will actually start the hemolysis at 37 degrees Celsius, and they will actually complete the hemolysis at 4 degrees Celsius. So that is hot at 37 degrees Celsius, cold at 4 degrees Celsius phenomena. So next method of identification is about the litmus milk. Heat tolerance I have discussed, the litmus milk. So the litmus milk means, so you can take a solution of this litmus milk, and here you are actually adding this Clostridium perfringens, and actually this alpha toxin, right? Actually this Clostridium perfringens, there is actually lactose. So that lactose will be fermented by this Clostridium perfringens, and by the fermentation it produces gas and acid. So this will actually this acid convert the casein into clot, and that will be actually uh, formed. It will be like a stomy. That means here and there. That is called the stomy clot reaction. In that way, we can identify the Clostridium perfringens. So the fifth method by which we can identify the Clostridium perfringens is by reverse camp test. So what is meant by this reverse camp test? So in this actually you have taken a blood agar as like this, and you have streaked the Clostridium perfringens and the Streptococcus agaricti in perpendicular. You have actually streaked. What you can observe is that there is actually more hemolysis, that is synergistic hemolysis, especially at the meeting region, and there will be like an arrow. The hemolysis will be like an arrow, and that will be actually pointing to this Clostridium perfringens. That is, the arrow will be pointing to the Clostridium perfringens. So that when you observe that, that is actually called the reverse camp test, and it is actually positive for the Clostridium perfringens. So next is an automated method that you can actually use, and that is multi-TOF. So that is all about the identification. Now I will be actually talking about the gas gangrene. So what is actually a gas gangrene? It is actually affecting the skeletal muscles. So it is anaerobic myonecrosis. Anaerobic, it is acting on muscles, so myonecrosis. So the organism, the species that is responsible for this gas gangrene is the Clostridium perfringens, and majority it is Clostridium perfringens. There are other Clostridial species which are also causing this gas gangrene. So it is actually progressive and rapidly acting, and it actually affects the skeletal muscles. So what is the pathogenesis behind it? So when there is a trauma, when we get a trauma, it can be during the road traffic accidents, or the during the wounds that we get during war, or during septic, during septic abortions. So when there is a wound, and the spores of this Clostridium perfringens, they manage to enter through the wound, and they will actually reach the vessels, that is a localized vessels. They actually damages it, and there will be resulting ischemia. So the tissue is ischemic. So, and actually there will be it actually disseminates through the blood, and as a complication of that, there will be clotting, because alpha toxin actually have the ability to cause platelet aggregation. So here and there will be clotting, that is disseminated intravascular coagulation, and many more complications. And actually the presenting feature will be, there will be a discoloration. As you can, the images actually you can refer to the Apurva Shastri. There will be discolorations, and it will be having severe pain, and there will be actually bullas, that is hemorrhagic bulla over the skin. And you can see that the air gets trapped in the muscle plane, and there will be actually crepitus in the muscles. And actually uh, when as there anaerobic medium, they gets an anaerobic environment, they goes on multiplying and multiply. This Clostridium perfringens is goes on multiply and multiply. So when multiples or actually bacteria are present at the same time, for example, if there is E. coli, Klebsiella, and certain facultative anaerobe, what happens is that the oxygen is going on reducing, because these organisms will use facultative anaerobes, so they all will use oxygen, and the oxygen goes on reducing, and it creates an anaerobic environment. So they goes on multiplying and multiplying, start to release their virulence factors, and cause all such complications. It can also cause multi-organ failure, and 50% death. There is actually 50% death. So it's actually a serious condition. So that is actually gas gangrene. Now about the spontaneous gas gangrene. So about spontaneous gas gangrene, actually there will be the portal of entry is not through the trauma. Actually there won't be visible trauma. So actually there will be underlying pathology of the intestine. That is sometimes there will be inflammation of the ileum, that is ileitis, or sometimes like colonic diverticulitis, or even there can be a colonic carcinoma, and the trauma, that is and actually through that they can these spores can enter, and it can hematogenously spread, that is through the blood it can reach the muscle. So that is about the spontaneous gas gangrene. Now about the another manifestation caused by the Clostridium perfringens, that is about the Clostridial food poisoning. So what is basically this Clostridial food poisoning? Actually, this Clostridium, it is having the ability to release enterotoxin. Actually, Clostridial perfringens enterotoxin. So there is a gene, CPE gene, that will produce this enterotoxin. And actually, when you are heating, when you are heating the food that is contaminated, especially the meat products that are contaminated with the spores, actually these won't get. Actually, when you are heating for 100 degrees Celsius, this for 2 to 3 hours, it can actually survive, and it actually in the intestine it can cause spores. Actually it can cause spores in the intestinal mucosa, and there will be actually and actually there will be watery diarrhea, then abdominal cramp. Actually the abdominal cramp. So actually the treatment is actually symptomatic treatment. Within 1 day it actually resolved by itself. And about the I have forgot to discuss about the treatment of the gas gangrene. So in the treatment of gas gangrene, what you can give is early surgical debridement. you can actually remove that, especially surgically. Uh, then after that, you can use antibiotics, that is IV penicillin and clindamycin. Then you can use hyperbaric oxygen. So more oxygen to that region means it actually kills those uh, Clostridium perfringens. And one more thing is that you can also give a passive immunization, that is anti-toxin, that is anti-alpha toxin anti-serum can be given. So another manifestation produced by this Clostridium perfringens is that they will actually cause this necrotizing enteritis. Actually, it is also called as pig bell. That is, so when you are actually eating the contaminated pig pork, when you are actually eating contaminated pork, so this actually contaminated, so especially which are whether Clostridium perfringens, actually type C, most commonly type C. Uh, it will actually the clinical presentation would be of dysentery, that is bloody diarrhea, abdominal cramp and fever. So on treatment what you can give is IV penicillin plus metronidazole. So that's all about the Clostridium perfringens. And one more thing is that you can actually cultivate this by using anaerobic jar. So, and you can also do it using anaerobic gas pack system. Remember that in these two we will use an indicator, and that indicator is methylene blue. So what the methylene blue will do is that, so the methylene blue will be in that container, and when there is anaerobic environment in the container, this methylene blue, it becomes colorless. That is how we will identify whether inside it is anaerobic or not. Then we can also use a sterile petroleum jelly. Even above the culture medium, we can add this sterile petroleum jelly so that oxygen diffusion is not there. So that is all about the Clostridium perfringens. Okay.